Swayam Prabha. Digital India. Educated India.
Welcome to the last but one class of this course. In my last lecture we were discussing about the passive immunity and their future trend, what can be done with the passive immunity. So, today what I am going to do is I am going to talk about antibody that we can generate in laboratory, because whatever the passive immunity is the antibody raised in other organism or other individuals, which we can use for the treatment or curing or the neutralization of the toxin, this for this kind of reason. But the thing is, the supply of antibody from animal, there are certain problems that we have discussed in the last class, but so we should have a system from where we can generate a huge amount of antibody, so that we can treat the patient for either say snake bite or any disease already like tetanus or rabies, which is already developed, because immunity or active immunity development we do not have that much time. So, we need a ready-made antibody in our hand, and that should be a good amount, and in animal definitely there are a lot of antivenoms we are still producing in horse. But if we have certain facility or the system where we can generate antibody without going to animal, will be better. So, before going to do the monoclonal antibody, let us discuss one more time what is what do you mean by, or what we understand about the polyclonal and monoclonal antibody. So, let us start. Like today we are going to talk about the number of lecture is lecture number 59. So, first let me discuss or start polyclonal antibody. Okay. Polyclonal antibody. Okay. So, you know what is clone? Clone is the same thing, multiple copies of the same or identical thing. This is called clone. So, polyclonal means there are multiple clones are there. How multiple clone can develop in a regular immune system or active immunity? Suppose this is the a protein antigen that we are injecting, and there are, say for example, 2 epitopes. So, this is one epitope and another epitope, say this region is the another epitope. So, why I am showing this? This is the linear epitope, the red one is the linear epitope and the blue one is the conformational epitope. Okay. So, now if this antigen we administer into animal, okay, or any pathogen attack, what will happen? This antigen will enter into our body or in any organism, and then immune system will react. Okay. So, immune system, what will happen? B cell receptor will recognize this epitope. This is I am talking about B cell epitope. So, red and blue are the blue cell epitopes. Okay. B cell epitopes. So, that B cell epitope, what will happen? So, suppose this this epitope is look like this, and the blue epitope is look like this. Okay. This is the blue epitope is look like this. Okay. So, this is the 3-dimensional structure. Now, what will happen? So, if this is there is antibody, antibody's receptor will be like this. Okay. Right. So, this is the B cell epitope. Now, if I delete this polyclonal part, I am talking or discussing about the polyclonal antibody. So, now, so this is B cell, and another antibody will be like this, which is again attached to it. So, this is again a B cell receptor. First the epitope will be recognized by the B cell, So, this is another B cell. Suppose this is B cell 2, this is B cell 1. So, in this I am just considering only one B cell reacting with red epitope, another B cell 2 is reacting with blue epitope. What will happen? Both the B cell ultimately will be converted to plasma cell and produce antibody. So, what we will see after 7 days or around 10 days if we see. So, there will be antibody like this. And from this B cell what kind of antibody will be produced? This B cell antibody will be like this. Right. So, this will be antibody. So, in serum both type of antibody will be there and both are going to recognize this antigen, because in antigen I do not know how many epitopes are there. It may be a single epitope, it may be multiple epitope, multiple means 2, 3, 4 depending on the size of the antigen. If the protein is very big, we will have multiple epitope. So, if there are 4 epitopes, at least there will be 4 different B cell will be activated, 4 different kind of antibody. So, in serum we will get the pool of antibody which comes from multiple B cell. So, this mixture when we get, this is called polyclonal antibody. Okay. This is going to make polyclonal antibody. Okay. But now assume there is only one epitope, suppose this blue is not there, only red is there. Even after that, if I inject that antigen into animal, what will happen? So, one B cell 1 will come here, another B cell may come which may recognize this same epitope, but it is slightly different in sequence. Okay. Slightly different in sequence, suppose in the epitope binding site, the paratope of the antibody or the B cell receptor, suppose in one B cell it is aspartic acid, in another B cell it is glutamic acid, rest of the sequence are same. So, what will happen? Both will bind, right. So, what will happen then? So, another B cell will come. So, assume that this epitope is not there, only one epitope is there. So, another B cell will come, say B cell 1 prime, okay, B cell 1 prime which has similar epitope. So, even suppose in that case both B1 B cell 1 prime or 2 B cell are going to produce a mixture of antibody which is almost identical, but they are originated from 2 different B cell. So, 2 different B cell means 2 different clones, their sequence are very similar, but 1 or 2 amino acids in the interacting region or the paratope region are different. Then also we will call it polyclonal. So, even antigen has only one epitope, still the animal or a whole organism will produce antibody from multiple clone. Okay. What is happening actually after presenting by the antigen presenting cells, say dendritic cells, T cell will be activated and all the B cell which is recognizing this epitope will be activated by that T cell. So, all of them are going to convert it into plasma cell and produce antibody. So, this is then polyclonal antibody. Okay, even there is only one epitope, there is a very tricky question sometimes. So, if one antigen has only one epitope, whether the antibody produced in any animal, whether it is a polyclonal or monoclonal, it is polyclonal because multiple B cells, so 1, 2, 3, 4, multiple number of B cell may be activated and give the pool of total antibody which is coming from or which are coming from multiple B cells. Now, then also if only one epitope is present, only one is present, then also it is a polyclonal if it is raised in animal. We have no control. Very unlikely that one epitope has only one B cell in body. So, even if it is that, we cannot make sure, right. It will be a lot of, you have to isolate, purify, sequence and then confirm. So, normally we say it polyclonal. Now, if any one of them, suppose there are 4 such or 3 such B cells which are producing antibody against epitope number 1. Okay. So, now if by any technique if we can identify this or separate this, and grow them in a culture or somewhere where all the B cells, okay, are of same type, say all are 1. Then what will happen? It will produce only one type of antibody. Then if this is, we can purify and culture in a system, so that we can get antibody from that, all antibodies are going to be identical. Then it is called monoclonal. Okay, that means the antibody is getting monoclonal antibody. So, antibody we are getting from a single clone. It also called MAB. Many times you will see MAB. So, this is monoclonal. Now, we are going to discuss how we can get monoclonal antibody. So, it was first discovered, or the technique was first discovered long back, it is 1975. Okay. George Kohler and Cesar Milstein. George Kohler and Cesar Milstein, who developed this technique, and they received Nobel for just developing this technique in 1984. So, they received the Nobel Prize for this technique. So, it is a very beautiful technique. Now many labs are producing that, but initially their idea was fantastic, what they did. So, now we will see what they actually did and how we can get the monoclonal antibody. Okay. So, let us see that I have one B cell. Now, how I will get one B cell? So, this is one B cell. Okay. How I will get one B cell? It is not possible. Normally what we do, suppose I have one antigen, say antigen X. Okay. I would like to make monoclonal antibody against this antigen X. So, what I have to do is, I have to take a mouse. Okay. So, this mouse I will inject this antigen X into it. So, what will happen? This mouse will be primed with this antigen X. So, if I would like to raise antibody in any animal, what I do? I just discussed in last class that we are going to use adjuvant. In case of animal, we are using Friend's complete adjuvant, that means oil in water emulsion plus it kill bacteria with it. So, first injection, then first booster, second booster with incomplete adjuvant, and to raise the antibody level high. So, what will happen if I inject antigen, say for 1 time, 2 times, 3 times, depending on how good your antigen is. So, after say second booster, if I will isolate spleen, because from spleen I will get many B cells which are already primed with that. So, what will happen if you take a mouse and directly isolate the spleen, you will get lot of B cells, but activated B cell against this antigen X will not be that many. So, as I am interested to develop monoclonal against antigen X, so better is you prime that animal, so the number of B cell activated against antigen X will be more. So, after you isolate the spleen, you can get the B cell from it. Okay. So, it is normally spleen cell in total called splenocytes, and spleen is a secondary lymphoid organ. So, we have lot of B cells, lot of T cells. So, here what will happen, you will have, you can purify or isolate the B cell in different way. So, you know the flow cytometry or the facts we can use at this moment. So, we will have, so I am making small B cells, now all are red. Okay. So, lot of B cells we will get. So, these B cells I do not know which one is, there are millions. So, out of these million cells I do not know exactly which one is actually activated and which one is against this antigen X, it is not known. So, suppose at the beginning we do not have to know, but I can say that the number of B cells against antigen X is better or more than if you take a mouse directly. Okay, if you take one mouse directly, then the number of B cell will not that many. You can pull multiple or more number of mouse also. Okay. So, suppose one mouse, you isolate the spleen, then from there you take B cells. So, these are already activated. So, if you grow them in cell culture medium, medium you know, medium is just a food source, like you, I am definitely all of you grown bacteria in LB or other nutrient bath. So, that gives the food and the basic materials of food. So, similarly there are medium specialized for animal cell culture. Okay, there are many, say RPMI is one, RPMI, Roswell Park Memorial Institute. This is a name of one institute in Buffalo, New York. They developed a series of medium. Okay. So, RPMI, there are different number depending on how they develop. One medium is there is another called Dalbeco, modified, Eagles medium, okay, DMEM. So, there are various kind of animal cell culture medium that the way we have in bacteria, like Luria broth, Luria bartini, and then nutrient broth, potato dextrose medium. So, these are the animal cell culture medium. But if we grow in this medium, what is going to happen? These cells has a limited lifespan. They will not grow forever. After certain generation, all of them will die. So, that is not a permanent solution. So, people tried that. So, then this Milstein and Kohler, they figure out how to make them immortal. Okay. So, what they did, they took another cell, okay, which is cancerous B cell. This is cancerous B cell, myeloma. Okay. These myeloma cells, they are B cells, cancerous, that means they are immortal. They are immortal, they do not die. So, they become transformed, or they after transformation they became immortal. But what they figure out, that should be some property. What? First, they should not produce antibody. So, myeloma cell they purify, which has the immortality, they are also B cell, but they can't produce antibody. So, then they thought these cells are able to or capable of producing antibody, but they are not immortal. These cells are immortal, but they can't make antibody, or they selected that way, so they are not making antibody. So, they thought if we put them together, okay, if we put them together and fuse them, just fuse them and select the cell which have both the property. That means, half property, that immortality will come from this myeloma, and antibody producing property will come from this B cell. So, what will happen? We will get a cell, so which will have half the property of myeloma, that means they will be immortal, okay, they will be immortal, and half the property of this B cell, that means they will produce antibody. So, how to do that? So, they make many such I mean they figure out what to do. You can do two different way. One, if you put them in electric field, okay, they will fuse. Basically, you have to fuse these two cells. So, B cell and myeloma cell you have to fuse and make them one, okay, having both the property. So, you can do electrofusion or can use PEG, polyethylene glycol, polyethylene glycol. PEG has the property, they fuse the cell. Okay. So, now this kind of hybrid cell is generated, and that is why this cell is also called, the technology also called hybridoma. Okay. Hybridoma technology, that was actually discovered. But now what will happen if you give PEG, both red cell will also fuse, right, to many B cell will also fuse, and similarly the green cell to myeloma cell will also fuse, because PEG will not selectively fuse only green and one green one red. Say they will make both fusion to many B cell will be fused like and make like B cell fusion. Some this is BB fusion, and this is myeloma fusion, and this is B cell and myeloma fusion, and we want this. So, in the mixture both myeloma fusion, B cell fusion and B cell myeloma fusion, we have to select only this one, because we do not need these two. For selection of this, it is not to get rid of this, it is not a big deal, because they are not going to survive after few cycles anyway, right, they are going to die. But the problem is to get rid of this myeloma fusion, because there will be a lot of after this experiment you have huge number of cells, okay, millions of cells. So, from there finding out the right one which will produce antibody against antigen X is one of the very tough job, okay. It is cumbersome, lot of work you have to do, and in that if you have a mixture of this myeloma, it will make the technique even or the system more tougher than the only B cell myeloma cell fusion. So, what we have to do? We have to figure out something so that they will not also survive. So, now we will put in a medium, only this cell will survive. For what they did? This myeloma cell was chosen in that way, they are mutant in AGPRT. AGPRT is an enzyme, hypoxanthine guanine phosphorybosyl transferase, HGPRT, hypoxanthine guanine phosphoribosyl transferase. What this enzyme is doing? This enzyme actually involved in nucleic acid biosynthesis. So, all the nucleotide synthesis, there are two pathways in every cells. Okay, in every cells in our body we have two different pathway. One is de novo pathway. I hope you already know this from your metabolism course in the biochemistry. One is de novo pathway, and number 2 is salvage pathway. Okay. So, de novo and salvage. This HGPRT is a very important enzyme of this salvage pathway. So, what is happening? This myeloma cell, as this major one of the key enzyme, that HGPRT is not there. So, they cannot use salvage pathway. Okay. Even but they do not care normally, because their de novo pathway is active. Okay. So, they can make their nucleotide and survive. But in this medium where we grow mixture of all this, because in after PEG you have this, you have this, you have this. Okay. These we do not care much, because they will die after few cycle of their life cycle, but this cell and this cell both will survive. So, we have to get rid of that. They do not have HGPRT, fine, but they have de novo pathway. So, what we do is, we in that medium we add hypoxanthine, we add aminopterin, and we add thymidine. So, hypoxanthine, thymidine, both nucleotide, they are going to use for their nucleic acid biosynthesis or DNA synthesis, but they can't use, the myeloma cannot use this hypoxanthine, because they do not have hypoxanthine guanine phosphoribosyl transferase. So, for them, for myeloma cell or the myeloma fused cell, so some this myeloma cell will also be there. So, in this mixture there will be 5 type of cell. What are these 5 type of cells? One is BB fused, some unfused cells of B type and some unfused cells of this myeloma cell type. Okay, so 5 type of cells. So, this fused B cell or unfused B cell will die, but both this myeloma and myeloma fused and hybridoma, all 3 will survive, but these 2 does not have a GPRT. So, they cannot use this hypoxanthine or salvage pathway, but we have to stop this de novo, so that they cannot synthesize their nucleotide at all. So, aminopterin, this is the inhibitor of one of the enzyme of de novo pathway. What is that enzyme? This is tetrahydrofolate, the folic acid. Okay. So, to stop their de novo pathway, we add one specific inhibitor called aminopterin. Aminopterin actually inhibits the de novo pathway by inhibiting one very important enzyme of that pathway. It is dihydrofolate reductase. Okay. So, what is, as a result, what is happening, they cannot use hypoxanthine or the salvage pathway, they cannot use the de novo pathway, because aminopterin is there as an inhibitor. So, both nucleic acid synthesis pathway, de novo and salvage, are blocked. So, what is happening with time, both these fused cells as well as single myeloma cell all are going to die, and these cell also are going to die by natural death, because they cannot survive more than few cycles. So, as a result, ultimately we will have only this hybridoma, where B cell and myeloma cell get fused. Clear? So, then what is happening? So, finally, after the death of both the fused myeloma and single myeloma cells and B cells, we will have only hybridoma cells. So, these hybridoma cells are, then we have to figure out which hybridoma cell is actually I need or we need. So, how we will select, because that hybridoma cell are going to produce antibody against this antigen X. Okay. Suppose there are 1000 such cells, there will be many. Suppose there are 1000 such hybridoma cells are there. Okay. So, from there we have to select how many of those are producing antibody of what I need or I want. So, I have to dilute in such a way. So, normally we use 96 well microtiter plate, okay, 96 well plate or microtiter plate. So, we will dilute in such a way, each well should have one cell, each well should have one cell. So, 1000 cell if we have, so what should be the dilution? You have to dilute it 1000 times. So, 1000 cell per ml, for example. So, we have to dilute in such a way, so that one cell per well. So, we have to dilute 1000 times, right. So, 1000 time dilution, and that dilution if we grow in individual cell. So, I have, not necessarily 96 well plate means I may have to take 12 96 well plate. Okay. So, each well I will give this cell in such a way. So, 1 microliter each. So, they all will get one cell. It is possible that some well does not get any cell, another well get 2 cells. Okay. Then I will grow for say 14 days. So, what will happen? From 1 they will increase, they will grow up to certain level, and during that growth, if I draw this in bigger way, in little bigger format. So, what is going to happen? Suppose in that this well is big. So, in that there are multiple cells. Okay. So, while they are growing, the hybridoma cells, all are hybridoma I am talking. Okay. So, while growing they will also secrete antibody. So, with growth they will release the antibody also, okay, in the medium. So, if we take the medium out, take some sample and take the medium out, and so what will happen in that medium, I will have only antibody. So, if I centrifuge the cell and collect the supernatant, I will have the, so centrifuge and then collect the supernatant. In supernatant, I will have only antibody. So, whether that antibody is against my protein of or antigen of interest or not, what I can do? You know that now. This is simple ELISA. Okay. So, if I do ELISA, I can, if the ELISA is positive, then I can say that is the real well from where my cell. So, say 1000 ELISA I have to do, because I distributed in 1000 wells and I have to do individually the product from each well. So, now you can imagine what is the volume of total work. So, if the number is more, if it is 10 to the power 6, we have to make so many plates. So, now this 1000 well I have to do 1000 independent ELISA. From there I may get say 10 out of 1000 are giving ELISA positive. So, this is the first round of test. Then what I will do? I will take that 10 cells and grow them again in different well, freshly I will grow. Again I will do ELISA. Okay. Then I will see maybe next ELISA, first time sometimes false positive may possible. So, next time if I get all 10 are again positive, that I am very lucky. Many times it happen only 2 are positive or only 3 or 4 are positive. So, from there I have to select the single clone. Okay. So, after first round of screening, I will, what I will do? I will dilute them again. Okay. I will dilute them again, so that by any chance what I was saying, in some well there are 2 cells, it is possible that in some wells there are 3 cells also. So, I do not know whether the positive cells are from product of 3 or product of 2. So, what I will do? From there I will second round I will dilute them again. Okay. So, suppose there are 2 cells. So, what will happen? And total again 1000 cell. So, probability or statistics wise there will be 500 and 500. So, if everything is equal. So, 500 cell is one type and 500 cell is of another type. Again if I dilute, so every 2 wells I can have a single clone. So, that way I have to make sure that this antibody what we are getting is actually from one B cell or one hybridoma cell. The advantage is, now once it is ready, then it will live lifelong, and as many cycle you want, whenever you want, what you have to do? Again you grow this cell. I mean, where we are growing? We are growing them in RPMI, that medium. So, whenever I need, I will culture them, increase the volume. Okay. There are varieties of bioreactors are available. Normally this case, the hybridoma cell line we grow hollow fiber bioreactor. Okay. We grow this cell in hollow fiber bioreactor. So, in that case we can increase the volume, we can produce more, and we can separate or purify the antibody, and that can be used for different purpose. And that antibody later on variety of techniques develop, we can make humanize, so that anti-idiotype reaction will not happen. Okay. Once you have the clone, then or the cell isolated, then you can clone the gene, that we will discuss in the next class. You can clone the gene, and that clone you can express in yeast, bacteria, plant. Okay. If we express the antibody in plant, there is a very nice name is there, it is called plantibody. Okay. What will happen? You grow the plant, purify the antibody from total plant protein. Okay. You do not have to go cell culture, you clone the gene in plant, make a transgenic plant with a particular antibody, and then it is not that easy, but possible. So, now once you have a monoclonal, you can do many things. You can maintain the monoclonal, purify the antibody, do your serve your purpose, or you can clone the gene and express in heterologous system like yeast, bacteria, other system like plant, and purify whenever you need. So, okay. So, there are few things like how we can make the antibody in lab by molecular biology technique, we will discuss in the next class. For till then I mean I will just what I would like to show one slide what I tell, because this course is almost going to be finished. Okay. So, in my lecture all the slides or most of the slides rather I used from this book, the Genoimmunobiology, and it is a copyrighted, but I am also thankful to Mr. Ross Davis, Sales and Marketing Executive of W. Norton company, who is now the authorized agent of this book and seller publishers. So, and they are kind enough to give me permission to use this all these materials for this course. I am very much thankful to them. Thank you very much.